Hello, my name is Gerald Nwosu, and I am a PhD candidate in the Qiong lab studying childhood onset developmental and epileptic encephalopathies in the neurology department here at Vanderbilt University Medical Center. Our lab is interested in investigating rescue mechanisms for GABA-A receptor and GABA transporter mediated epileptic disorders. We have recently published a study in Brain Communications titled 4-phenylbutyrate restored GABA uptake and reduced seizures in SLC6A1 patient variants bearing cell and mouse models. In this study, we used various model systems to investigate GABA transporter function in conjunction with administration of a co-chaperone 4-phenylbutyrate. 4-phenylbutyrate, also known as 4-PBA, is a small molecule that has been used for treatment of urea cycle disorders in children. 4-PBA has also been reported to be beneficial for other conditions, such as cystic fibrosis, liver injury, ischemia, and Alzheimer's disease due to its ability to reduce the burden of mutant or misfolded proteins and ER stress. SLC6A1 encoding GABA transporter 1 has been associated with epilepsy, autism, and neurodevelopmental delay. We have identified that reduced GABA uptake, a common pathophysiology for all associated mutations, is caused by a destabilized protein, protein trafficking, and endoplasmic reticulum retention of the mutant protein. In this study, we hypothesized that the pharmacochaperoning effects of 4-PBA could restore GABA uptake function through stabilizing the mutant protein and trafficking more functional protein to the cell surface. We used several cell and knockin mouse models to evaluate the effects of 4-PBA use. As GAT1 is expressed in both astrocytes and neurons, we used both cultured from variant-bearing knockin mice and human patient-derived astrocytes and neurons. We evaluated the effect of 4-PBA on 8 missense and nonsense variants associated with heterogeneous clinical phenotypes. Each variant showed a reduced GABA uptake ratio in comparison to wild-type protein. This was corroborated with a low surface and total fluorescence intensity for each of the variants, excluding D410E and L460R in the total fluorescence intensity condition. Similar to the patient condition of heterozygous mutation, we tested the effect of PBA in cells expressing a mixture of the wild-type and mutant cDNAs to demonstrate the heterozygous condition, and mutant cDNAs alone to demonstrate the homozygous condition for comparison. Taking the ratio of tritium GABA uptake, we were able to assess the differences in GABA uptake for each variant as a result of PBA administration. Only variants E16 and L460R did not show a significant increase in GABA uptake as a result of PBA administration in the homozygous condition. However, under the heterozygous condition seen in clinic, there was a significant increase in GABA uptake across all variants. Additionally, we evaluated the effect of 4-PBA in a knockin mouse model. A major phenotype in SLC6A1 variant-mediated disorders is epilepsy, leading us to investigate whether PBA reduces seizure activity in mice. We tested the effect of PBA in SLC6A1 S295L mutant mice. After a 5 to 7 day recovery period following EEG electrode implantation, mice were treated with saline vehicle for 7 days and then subjected to video-monitored EEG recording for 48 hours to gather baseline EEG activity. After a day of rest, the mice were administered PBA for 7 days, followed by 48-hour EEG recording. Mice displayed frequent absence-like activity within the 5 to 7 Hz frequency band in baseline recordings. However, PBA treatment reduced the 5 to 7 Hz spike wave discharges from approximately 127 to 33 over a 48-hour recording period. This suggests that PBA can reduce seizure activity likely due to promoted membrane protein forward trafficking and increased functional GAT1 expression. We are grateful to all previous trainees, postdocs, students, and collaborators who made a wonderful contribution to this work. If you would like to know more about this study, please see the whole paper in Brain Communications.